Welcome to AVT2032 Principles of Animal Healthcare and Disease. Today we are going into a new topic 2.1. Veterinary pathology is the study of disease in animals, focusing on causes, mechanisms, and effects. This topic will explore the definition of pathology, its branches, and essential terminology related to disease conditions, providing a foundational understanding for further exploration. Pathology is the study of disease, focusing on its causes, development, and effects. Therefore, the learning outcomes for this topic will be: define pathology, describe the branches of pathology, describe appropriate terminology in relation to disease conditions. Before we go through learning outcomes, what is important to study pathology? Whether it's human or animal, the use of medicine is paramount because it is the cure and live healthy. So, we must study diseases. How this is achieved? By explaining the causes and development of disease. Pathology provides the scientific foundation for the practice of medicine. Also, pathology is the bridge between science and medicine. Pathology is essential for understanding disease, serving as the scientific basis for medicine. It bridges science and medicine by examining disease effects at multiple levels: whole body, organs, tissues, cells, and subcellular structures. This comprehensive study aids in diagnosing and treating diseases effectively. What is pathology? Pathology is branch of medical science, study of the causes of diseases, how they develop and their effects on the body. In terms of veterinary pathology, it defines the science that studies the causes and effects of disease in animals. Etiology, study of science of the causes of diseases. Pathogenic, a factor which is capable of producing disease, can be referred as pathogenic. Agar bacteria, virus, and fungi pathogenesis relates to things which produce disease and tends to be used when discussing how factors lead to diseases or the mechanisms of disease development. Pathology is crucial for diagnosing diseases and identifying causative agents, aiding in effective treatment. It benefits human health by addressing zoonotic diseases and enhances research by understanding disease progression and transmission. Additionally, pathology supports pharmaceutical research, aiding in drug development and treatment methods. General pathology focuses on the fundamental mechanisms of disease, such as etiology and pathogenesis, without limiting to specific organs. For instance, inflammation is a cellular response to injury. Systemic pathology delves into these processes with a focus on specific tissues or organs, examining aspects like etiology, pathogenesis, and diagnostic features, exemplified by dermatitis, and inflammation of the skin. Anatomic pathology focuses on diagnosing diseases through the examination of organs and tissues, utilizing techniques like histopathology and autopsy. Clinical pathology aids diagnosis via laboratory tests on blood, urine, and other fluids, with specialties such as hematopathology and clinical microbiology. Both branches are crucial for comprehensive disease understanding. To effectively study disease conditions, use a consistent format. Start with an overview, identify the pathogen or etiological agent, and understand its habitat and transmission. Delve into pathogenesis, observe clinical signs, and learn diagnostic methods. Finally, explore treatment options and prevention strategies to gain a comprehensive understanding. As we know, etiology is a cause or origin of disease. It depends on two factors: one, internal, and two, external factors. Internal factors divides into gene abnormalities, mutations, and deletions. In general, it's based on the gene or DNA level. Sequence variations. Second is chromosome abnormalities like presence of extra chromosomes. Third is the immune system, abnormal or defects. For example, we have seen this condition called immunodeficiency in topic 1.4. Finally, aging, natural process or premature aging. A lot of research is ongoing in this field, anti-aging. External factors influencing disease include physical agents like trauma and radiation, chemical agents such as toxins and acids, and infectious agents like bacteria and viruses. Environmental factors, including nutrition, temperature, and hygiene, also play a crucial role. Understanding these factors is essential for comprehending disease etiology and prevention strategies. Etiology refers to the cause of a disease. One etiology can lead to a single disease like canine distemper or multiple diseases such as Cushing's syndrome causing bacterial dermatitis and diabetes mellitus. Conversely, multiple etiologies like excess sugar intake and obesity can result in a single disease such as diabetes mellitus. Infectious diseases in companion animals include canine distemper virus, canine parvovirus, feline calicivirus, and feline herpes virus. Non-infectious diseases encompass obesity, cardiac diseases, osteoarthritis, and corneal ulcers. Understanding the distinction between infectious and non-infectious diseases is crucial for effective prevention and management strategies in veterinary care. Pathogens, including viruses, bacteria, fungi, and parasites, disrupt animal health by invading host cells, evading immune responses, and causing tissue damage. Pathogenicity depends on factors like virulence, transmission, and host susceptibility. The host's immune system responds through innate and adaptive mechanisms aiming to neutralize and eliminate these infectious agents, maintaining health and preventing disease progression. Symbiosis refers to the evolved interaction or close living relationship between organisms from different species. These relationships can provide benefits to one or both individuals involved. Understanding symbiotic relationships is crucial in studying ecological interactions and the balance of ecosystems. Symbiosis manifests in three forms: mutualism, commensalism, and parasitism. In mutualism, both parties benefit, as seen with plants and pollinators like bees. Commensalism benefits one organism without affecting the other, exemplified by barnacles on whales. Parasitism, however, benefits the parasite at the host's expense, such as Saculina carcini affecting crabs. The likelihood of contracting a disease depends on several factors. The animal's health and immunity status play crucial roles. Additionally, the pathogen's virulence, which refers to its toxicity and invasiveness, significantly influences disease contraction. Understanding these factors is essential for assessing disease risk and implementing effective prevention strategies. A pathogen is any agent capable of causing disease. Pathogenicity refers to the ability of an infectious agent to cause disease, while pathogenic describes something that causes disease. Understanding these terms is crucial for assessing disease risk and management strategies. Robert Koch in 1877 established 4 postulates to determine pathogenicity. These criteria help identify if an organism is the etiological agent, meaning the causative agent of a disease. Understanding these postulates is crucial for identifying and confirming the role of pathogens in disease causation. Koch's postulates require that the agent be isolated from every patient with the disease and propagated in pure culture. When introduced into an experimental host, it must reproduce the original disease. Finally, the agent should be re-isolated from this experimental infection, confirming its role as the causative agent. Koch's postulates are foundational in identifying disease-causing agents. They require the agent to be isolated from every patient, propagated in pure culture, and cause the original disease in an experimental host. The agent must then be re-isolated from this host, confirming its role in the disease process. Koch's postulates face limitations when organisms cannot be cultured, lack animal models, or have gained virulence. Subclinical infections and those affecting immunocompromised patients also challenge these postulates. Additionally, organisms accessing deep tissues through trauma or medical interventions complicate the application of Koch's criteria in identifying pathogens. Effective transmission of infectious diseases involves understanding modes and routes. Attachment to host surfaces is crucial, often facilitated by bacterial adhesins binding to host receptors. This process can be hindered by normal flora, mucosal antibodies, and antibacterial substances like lysozyme. Escherichia coli pili exemplify this attachment mechanism, highlighting the interaction between host and agent. Pathogens evade host defenses through firm attachment and repulsion of macrophages, often using capsules or cell walls to prevent phagocytic digestion. Dissemination occurs via blood, lymph, and other pathways aided by bacterial enzymes like collagenase and hyaluronidase, facilitating direct extension into surrounding tissues. Pathogens exploit host resources to thrive, manipulating the microenvironment to suit their needs. They adjust temperature, pH, and oxygen levels and absorb essential minerals like iron, ensuring optimal growth conditions. This strategic adaptation enhances their survival and proliferation within the host, complicating the host's defense mechanisms. Physical barriers, epithelial layer that covers the outside of our body and lines lumens, cavities, and spaces, is the first barrier or defense that must be overcome by a microbial infection. Chemical barriers, salivary secretions, mucus secretions, tears, gastric secretion, and vaginal secretions, natural killer cells, type of lymphocyte and component of innate immune system, play a major role in the host rejection of both tumors and virally infected cells. Acute inflammation is the initial response of the body to harmful stimuli and is achieved by the increased movement of plasma and leukocytes, especially granulocytes, from the blood into the injured tissues. Complement cascade is a biochemical process in the blood that helps or complements cells of the immune system to eliminate invading pathogens. Humoral immunity, mediated by antibody molecules that are secreted by plasma cells, involves B cells that recognize antigens or pathogens that are circulating in the lymph or blood. Cell-mediated immunity is an immune response that does not involve antibodies, but rather involves the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. Innate immunity. Innate immunity refers to non-specific defense mechanisms that come into play immediately or within hours of an antigen's appearance in the body. These mechanisms include physical barriers such as skin, chemicals in the blood, and immune system cells that attack foreign cells in the body. The innate immune response is activated by chemical properties of the antigen. Adaptive immunity. Adaptive immunity refers to antigen-specific immune response. The adaptive immune response is more complex than the innate. The antigen first must be processed and recognized. Once an antigen has been recognized, the adaptive immune system creates an army of immune cells specifically designed to attack that antigen. Adaptive immunity also includes a memory that makes future responses against a specific antigen more efficient. Successful infections can directly damage the host through toxins, viruses, and parasites. Endotoxins trigger cytokine release, causing fever, inflammation, and hemorrhage. Exotoxins, such as hemolysin, collagenase, and hyaluronidases, destroy cells and disrupt biochemical reactions by entering cells. Viruses can alter cell function and structure, leading to cell destruction. For instance, the canine distemper virus and feline panleukopenia virus target lymphocytes. Additionally, viruses may induce tumor formation. Parasites cause physical damage and disrupt normal functions, further complicating the host's health. Successful infections can lead to acute death, chronic infections, or subclinical conditions where symptoms are absent. Some infections result in slow recovery, impacting the host's health over time. Understanding these outcomes is crucial for effective disease management and prevention strategies. Let's end this topic 2.1 by watching a video in the next slides. WAHIS, the World Animal Health Information System, plays a crucial role in safeguarding animal health globally. It provides real-time data on animal diseases, enabling timely interventions and policy making. This system is vital for preventing disease spread, ensuring food security, and preserving biodiversity, ultimately contributing to a sustainable future.
Since its creation in 1924, one of the missions of the OIE has been to observe and analyze the status of animal diseases throughout the world. This is vital both for animal health and human health, as 60% of pathogens which affect humans are of animal origin. This is also the case for 3/4 of emerging infectious diseases appearing for the first time. Diseases such as avian influenza H5N1, rabies, tuberculosis, the Ebola virus, or foot and mouth disease pose serious threats to animal health and welfare, and also to human health. Today, the danger is becoming even greater. Because of globalization, infectious diseases can spread with lightning speed. Therefore, to prevent the spread of serious animal and human epidemics, it's essential to detect potential animal sources. This identification means disease can be controlled and people and animals protected. For this, there is a valuable tool, WAHIS. WAHIS is the Global Health Information System of the OIE, the World Organization for Animal Health. The system is built on several pillars: permanent surveillance on the ground, early detection of animal diseases, highly reliable information through scientific verification, and a fast and efficient system for information dissemination via the internet. Let's take a look at how WAHIS works. The first step is the collection of health information by veterinarians, with the support of people on the ground. Farmers, hunters, fishermen, and many others. Then the national veterinary authorities analyze the data and report the status of the disease to the OIE. This notification is mandatory for all 180 member countries linked electronically to WAHIS. Official data are analyzed and organized by OIE specialists. In parallel, the organization tracks and verifies rumors and other unofficial information to supplement official data after consultation. The second step is the dissemination of this information. It's done via an open access web interface, divided into 3 parts. A monitoring system provides the global situation of about 120 diseases of terrestrial and aquatic animals, both domestic and wild. An early warning system displays real-time notification of any potential health risks, including emerging diseases. WAHIS also summarizes information for each country, such as the animal population, the material and human capacity of veterinary services and laboratories, vaccine production, along with much more complementary information. Aware of the increasing demand, the OIE is constantly improving the WAHIS system. Firstly, by helping members improve their system of surveillance and reporting. Secondly, by facilitating the collection and dissemination of information through new technologies. And thirdly, by adding genetic information relating to pathogens to the existing database. Through an increasingly precise knowledge of the global health situation, every day WAHIS is playing its part in making our health policies more effective and making our world a safer place. For us today, for our children tomorrow. Because protecting animals means preserving our future.
Let's have a summary of what we have went through. Pathology explores animal diseases, focusing on general mechanisms and systemic impacts on organs. Key terms include etiology, pathogenesis, transmission routes, clinical signs, lesions, and sequelae. Differentiating infectious from non-infectious diseases is crucial, with numerous infectious causes. Koch's postulates help identify disease-causing agents.